Hello, and welcome to Alexander Disease Research Update, Episode 23. I'm Albee Messing from the Waisman Center at the University of Wisconsin, and this is a recording of the talk that I gave this past weekend at the Alexander Disease Conference in Philadelphia, August 3rd, 2024.
First of all, let me make disclosures, or non-disclosure really. I talk to Ionis, of course, a lot, but I don't get paid by them for anything, and I'll never make any money off of ASOs. So let's start out with some positives. From the first case report by Stuart Alexander in 1949 until the first actual meeting like this dedicated to Alexander disease, it was a period of 59 years. And that meeting was sponsored by the ULF and held in DeKalb, Illinois. So after that interval of 59 years, it was only 10 years until the next meeting, which was in Madison, and now it's only 6 years until we're here in Philadelphia. So I'm certainly hoping that that trend is continuing. And like Jenny mentioned at the beginning of the meeting this morning, maybe even we'll get to the point where we have enough to say to each other to meet every year, and a lot can get done. There are still other signs of progress. This graph shows the number of publications on Alexander disease in each year since 1949. And you can see it was sort of ambling along at a low rate until it's picked up about in the 1980s, and then it's starting this pretty steady rise. And I'm hoping that will continue as well. This is probably the right time for me to acknowledge the various people who have supported the research in the Wisconsin lab, of course the NIH, ULF, the Palomaro and WANMA funds, the Alter Ricard Fund, Lisa's Corner, Klein, and Google. And certainly the person who's been most responsible for moving that work forward in my lab has been Tracy, who joined the lab, I think in 2002, or thereabouts. 1999? Okay, so I think about this long period of research on Alexander disease as being in four distinct phases. First, there was this period from the first case report in 1949 for about 50 years when the disease was being defined, mostly by neuropathologists at the beginning, but culminating in the work by Mario Vanderknap and Jim Powers on the classic MRI criteria for diagnosis of Alexander disease. And then there was the genetics period, which is actually fairly brief, and then that prompted us immediately pursuing, trying to understand mechanisms and, of course, therapeutics. You know, I must say, for those of us who were working on Alexander disease during this early phase, when not much was going on, I think many of us probably shared the sentiment that was voiced by this child about science in general. The skull has 22 bones in it. I know at least one person who claims this is interesting. You know, we would be sort of looking around wondering if anybody else thinks this is an interesting problem to work on. We hoped that was going to be true, and you just keep going. Of course, all that changed when Mike Brenner and I, along with a few other people, identified GFAP as the causative gene for Alexander disease. And I think Natasha or Amy or both might have shown a diagram similar to this before that shows the GFAP protein as this structure, vertical structure in the center, and each of those small circles represents an individual Alexander disease patient. And one of the striking things about this, first of all, is we still believe that GFAP variants account for over 90% of the cases of Alexander disease. We're still not sure what's going on with the others, but of those GFAP variants, they are distributed throughout almost all the domains of the protein, and there are quite a number of them. It was such a clear result that it immediately became a standard of practice to do GFAP testing as a diagnostic test. Even before we had published it, it started moving into clinical labs. So I think the burst of research on genetics really occurred in that 5-year period between 2001 and 2005. Although we still see that new variants are getting published. And this just shows the new variants per year, and it's holding pretty steady. So just realize the major form of GFAP is 432 amino acids long. So far, there are 91 of those amino acids have been associated, or variants of 91 have been associated with Alexander disease. And each amino acid could conceivably change in a couple of different directions. So my last count was that there are 151 different variants. Plus, there are 15 or 20 other more complex changes, such as insertions or deletions. So there really are quite a variety of variants to contend with. Let's talk about the present then. Where are we now, and especially this mechanism era? And what we've been very fortunate with colleagues in this endeavor is to develop a variety of model systems to use to study how GFAP is having these effects. And the model systems range from the type of in vitro assembly that Natasha showed you a picture of before in Roy Quinlan's lab, the Drosophila models in Mel Feeney's lab, a variety of cell culture models, both in Roy Quinlan and Jim Coleman, and now many other labs, and the rodents in our lab. So these model systems have really been critical to trying to tease out the mechanisms. And we've learned a lot, but I have to say that we've learned so much that we're still trying to make sense of it and trying to understand what are the most critical aspects. So the basic question is, what happens after you get expression of a GFAP variant? And of course, the gene gets made into RNA, that gets translated into protein. That then quickly leads to an increase in protein, and that's part of the problem. It's not the entire problem, but it's part of the problem. And at some point along the way then, in the green box, we've identified a variety of changes that now occur in the astrocyte. There's increase in a number of factors, lamin A's, which are an intermediate filament in the nucleus, P53, iron, STAT3, which is a transcription factor. All of these things are increased. There's also decreases in a number of cellular functions, such as protein degradation, or at least this protein degradation machinery known as the proteasome. There's a drop or almost a complete absence in the rodent models of this phenomenon called adult neurogenesis, which occurs in just specific areas of the adult brain where you get continued generation of neurons. And that's coming from a stem cell that expresses GFAP. And when you have these GFAP variants, it's just wiped out. And then there are decreases in genes that we think are important for things like seizure control, glutamate transporter, known as GLT1, and potassium channel, known as KIR4.1. So there's all these things happening in the astrocyte, as well as then things happening outside the astrocyte, activation of microglia, and ultimately, of course, dysfunction of oligodendrocytes, where you then get problems with myelin. So there are lots of things going on. At the same time, we see activation of what we think are protective stress response pathways that could conceivably be manipulated in the future. So there's so much going on here that actually I think it helps explains the appeal of the simplicity of the ASO approach, where if you just prevent the GFAP from being made in the first place, it takes care of all of these downstream problems. And whether that works as well as we hope it will, remains to be seen, but we'll talk about that in a minute. Let me just offer what I see as just a few of the highlights, I think, from this era from 2005 to the present. First of all, in clinical research, I think far and away the most important thing that happened was being able to move beyond this age of onset classification system that had been in place since the 1970s, and nobody was ever completely happy with it, but it didn't really know how to fix it. And then in 2011, Adeline Vanderveer's group at CHOP, as well as Yoshida's group in Japan, came up with alternative proposals. So they're clearly better than the age of onset classifications, but there's still no real consensus about what the best way to group patients will be. Whether there are 2 types, 3, 4, maybe 9 types, we still don't really know. And we don't really know what the value is of continuing to make those subdivisions. So I think there's a lot that's going to be accomplished ahead of us in that. Secondly was the proof, really through the rodents, that engineering a variant in GFAP spontaneously leads to an increase in GFAP expression. So we, in experimental models, usually the way you would get increased GFAP would be to injure some other cell type. And then you get secondary activation of the astrocyte. So here we know that the problem began within the astrocyte, and this was an exact genetic mimic of the human disease. Third, I think the development of the Drosophila models in Melfini's lab offered up a whole wealth of genetic resources that made possible large-scale genetic screens. And that allows us now to identify a whole slew of potential modifiers of the disease phenotype that offer us a vast array of opportunities for studying ways to modify the disease course in the future. Fourth would be what Natasha just talked about. We have to think beyond just the sequence, the amino acid sequence of GFAP, to all of the changes, the post-translational changes that take place at many of those amino acids, such as phosphorylation. And those could be really critical for not only interpreting the disease state in an individual, but also pointing out therapeutic targets. And last, of course, would be ASOs as a promising treatment strategy for the disease. At this point, let me go on to ask what can we expect and what do we really want from research? Obviously, the first thing I think everyone in this room wants are treatments or cures. So there are a couple of ways to think about that. First of all, in the short term, all we have to talk about really is the ASO trial. And here, I want to stress that what I'm going to say is my personal opinion only. For the ASO trial, of course, the results won't be known until sometime in 2025. But it's not too early to be thinking about what might happen if there are certain outcomes. So what if it works? Of course, we're going to be thrilled if it works. But it may work better for some people than others. And I think there's going to be a lot of additional refinement of that treatment strategy that will really make it useful. Maybe different people need different doses. Maybe you don't need lifelong therapy. Maybe at some point you can be weaned off it. We don't really know. And so I think that if it works, and we find that out in 2025, there's still going to be, that opens up a whole area of study for optimizing the ASO strategy. Of course, the second possibility is, what if it doesn't work? And if it doesn't work, I think it's going to be really important to try to understand why it didn't work. The preclinical data in the cells and the animals that led up to this were incredibly strong. And so we really need to understand what happened if that didn't get translated into the human application. And so I'm certainly hope that we'll be able to continue with that. Longer term, clearly I think ASOs are the first treatment strategy that's going to be tested, but it's not going to be the last. And there are others already in development, and we can certainly look forward to those. Some of them you've, I think, already heard about. Viral vector gene therapy approach that's being done by Jinji and Guangping Gao at the University of Massachusetts, as well as others. So I think there is certainly going to be a lot of new treatment strategies being investigated. Let me talk very briefly then about what do we really want from research beyond just treatments and cures. And on the clinical side, I think we clearly will want a more refined definition of the disease. And many times we talk about all the different variants, the 151 variants in GFAP, all being more or less interchangeable. They all cause Alexander disease. At least we think they all cause Rosenthal fibers. But the reality is they're clearly not all the same. And we know that some variants cause a really severe course. Some variants cause a much milder course. And right now we have no understanding of why that is. And so really improved phenotyping of the disease is going to be really critical for trying to tease all of that out. But I also agree really with Natasha that fundamentally it's a disease of molecular and cell biology. And the future opportunities for treatments or cures are going to come from better understanding of the fundamental mechanisms of disease. And what I put up here, just by way of illustration, is just one corner of a figure from one of Tracy's recent reviews, just to give a visual impression of complexity. There's a lot going on. I've said that before in the astrocytes, and we just need to put a lot more effort into trying to understand them. Now, one fortunate aspect about needing to understand more about fundamental mechanisms is that there is this notion of a reactive astrocyte. And fortunately, there's growing interest in what it means to be a reactive astrocyte. This is a review article that was published in 2021, had 81 authors on it. I'm somewhere in the middle because after the first author, we were all alphabetized, so M was just right smack in the middle. And as a result of that, you can also see there are a number of publications in very high-profile journals, all having astrocyte reactivity or reactive astrocyte as a central theme in these papers. Some of these are review articles. Some of them are also studies where they're using in vitro or in culture platforms for reactive astrocytes to try to screen for chemical modulators of that reactivity. And these could be explored for Alexander disease. So another question is whether we need more model systems. We've got several already. There are some that are under development and some that people talk about. The question is, do we need them more? I'm not quite sure. One of the classic model organisms that's often used in genetics is C. elegans, a nematode. So far, certainly nobody's published anything on a C. elegans model of Alexander disease. There are a number of labs working on so-called organoids. I think they have potential. They also are a long ways from being validated as models of disease. And whether they're really going to tell us anything fundamentally new about Alexander disease, I just don't know. But I think something that is going to be critical is that we continue to pay attention to what's going on with other neurodegenerative diseases. This is just a graph from a recent paper on blood and CSF biomarkers for Alzheimer's disease. And actually, GFAP going up is one of the earliest changes you see in Alzheimer's. So there is the idea out there that's not widely accepted at all, but it's at least being explored, that it's the rise of GFAP in Alzheimer's that is driving some of the changes in that disease. And so that's going to be obviously a common element with Alexander's. And then we've also published on another protein called TDP43, which then links the Alexander disease in with disorders such as amyotrophic lateral sclerosis or some of the frontotemporal dementias. So, these neurodegenerative diseases are certainly interesting from a mechanistic standpoint. The more we can understand the overlap between them. But also some of them, like Alzheimer's, have much, much more money and resources going into them. So what we want to do is take advantage of all the work that they're doing on those diseases and apply it to Alexander disease. What I want to finish up with, though, is if anyone really is able to develop sufficient resources for funding research, what we really need is what I would call an investigator-initiated grants program. That's the model that at least was followed for most of the history of the NIH, and I think it served medicine very well. Over the years, I've heard that some people have expressed surprise that all of this work on Alexander disease came out of veterinary medicine. But that isn't even true. It came out of, first of all, experimental neuropathology as the main discipline. But that's only the half of it, because from the beginning, I began working with Mike Brenner on GFAP in 1989, and he was involved in almost all the Alexander work up until very recently. And Mike began his career as an assistant professor at Harvard, working on the developmental biology of slime molds. So I think we really have to be open to new ideas and where those new ideas might come from to really make the most rapid progress in dealing with Alexander disease. So investigator-initiated grants programs would be the goal. And I guess what I'm trying to say here is that we shouldn't be telling people what we want them to work on. We don't want to be very specific about it because they may have a better idea. And unless you announce this sort of grants program, people won't realize that there's resources available to work on it. Let me finish up with a few comments about what only patients can do. And the first thing is continue to participate in research, like in the natural history study that's going on, and perhaps other things that are going on at other institutions. The second thing would be continue to provide donations of tissue, such as blood and CSF. And the more we can develop repositories of those samples and then tie them in to the clinical states of the individuals that they came from, the more valuable they will be. The last thing is I'll bring up something that some people may find unpleasant, but I think is really important. And the last thing that you could do is allow for autopsies. It's your last chance to make a contribution to research, and it's a gift to the future. And believe me, the future needs it. So thinking about autopsies ahead of time would be a great thing for the future of Alexander disease research. I think we have a lot of challenges ahead of us, that's for sure. But let me just close with this comment from 2,500 years ago, from Lost Play by Euripides. Nothing is hopeless. We must hope for everything. I completely believe this, and I hope you do too. Thank you.